Hello students. In previous lecture we have discussed about cell cycle and today we are going to discuss about M phase of a cell cycle, and that is also called cell division phase, in which we will discuss in detail a cell division phase can be divided into two groups, a karyokinesis and cytokinesis. So today we are going to start karyokinesis. The karyokinesis stage is divided into four substages, and out of that today we are discussing two stages, and that is prophase and metaphase. So today we are going to discuss about M phase of a cell cycle. Previously we have already discussed about interphase of a cell cycle, in which G1 phase we have learned, S phase and G2 phase, and after the G2 phase M phase will be occur. Okay, but that M phase may be divided into two groups, that is a mitosis and meiosis. So let me differentiate between mitosis and meiosis. What are the differences between mitosis and the meiosis? So here you can see this is a diploid number of chromosomes. Okay, this is a 2n number of chromosomes, and the chromosome which are obtained in the offspring or daughter cells will also have a diploid number of chromosome. But here what happened, a number of chromosome will be diploid in case of a meiosis at the initial level, but after the division, after two-time division, it will be converted into haploid structure. So here what happened, parent cell itself is a diploid and the daughter cell also diploid. Okay, whereas in this case what happened, a 2n, that is a diploid parent cell, will generate haploid daughter cells. So this is the main difference. Now let me understand how it is differentiated. It means when this mitosis occur and when this any organism will have two type of cells, and that may be divided into two groups, somatic cells and gametic cells. Okay, so let me understand what is the somatic cells and what are the gametic cells, that is also called gametes. Okay, so somatic cells are the cells in the body except for the sperm and eggs. So whatever the cells of rest of the body, okay, except the reproductive cells, okay, that is our sperm cells and the egg cell, all other cells other than the sperm cells and egg cells which are participating in the reproduction, other than that cells are considered as the somatic cells. Suppose a cell from our nose, cell of our cheeks, okay, skin, liver, all that are somatic cells. Okay, only cells which are participating in the reproduction, they are calling as gametes. Okay, and they are always diploid, means somatic cell will always diploid and produced by mitosis. So for the production of somatic cells, mitosis play very important role. So somatic cell reproduction or somatic cell generation will be occur by using mitosis process. So how gametes will be generated in males? Okay, only we understand that males will produce sperm cell. In case of a plant, they will produce pollen cells. Okay, these two are reproductive cells. Okay, and we are calling it as a gametes, male gametes, and female will produce egg cells. Okay, that is a female gametes. These all type of gametes, whether it is in case of animal it is sperm cells, and in case of a plant cell it is a pollen cell. Okay, and in case of a female it is considered as the egg cell. So these all cells which are participating in the reproduction are called gametes. Okay, and they are always haploid. Okay, means n number of chromosomes will present. Here 2n number of chromosome will present. Okay, so there, if haploid cells are there, then these will be produced by using meiosis process. Okay, so we have to very clearly we have to understand when mitosis will occur, each cell is a somatic of kind. Okay, then and suppose it will be having a diploid number of chromosome, so they will be produced by the mitosis. And if they are gametes and formation of gametes are the process, then it will be a haploid structure. So if you want to produce a haploid cells, then meiosis will play a very important role here. We understand more by using this diagram. Here you can see somatic cell division is there. That means they are reproducing or produced by the mitosis. So initially the cell will be in the G1 phase, cell will be diploid. Here you can see this is a 2n. Okay, then in the S phase what happened? A chromosome number will be remain same, but the DNA content will be double. Okay, so 2n become 2n only. Okay, means it will be not number of chromosome will not increase, the chromatin body will be increase. Then G2 phase, double the content of the DNA will be found, and then what happen in the after the M phase, okay, both the cell or nucleus karyokinesis, by using karyokinesis, will be divided and two daughter cell will be formed and they will have a 2N, and they're the diploid daughter cells. Okay, and in case of a meiosis, what happen? Initially cell will be a diploid. Okay, here you can see 2N, but after the meiosis, what happened? In the first meiosis, second meiosis, what happened? These 2N or a diploid cell will convert into haploid. So chromosome number will be half. Okay, this is the main phenomena of this process. Here chromosome number will be remain same whether it is parent cell. Okay, from the parent cell same number of chromosome will be obtained for the daughter cells. But here what happened? From the parent cell half number of chromosome will be obtained. So today we are going to discuss in detail about the mitosis, that is a somatic cell division process. So it is a division occurring in the somatic cell. Now you are very clear what is a somatic cell and what are the gametes. Okay, it is also called equational division. Why it is called equational division? Because same number of chromosome will be found in the daughter cell. If you compare parent cell and daughter cell, both cell will have same number of chromosome, and that's why it is called equational division, as the number of chromosome in the parent and progeny cells are the same. Mitosis is generally seen in the diploid cell. Okay, it also occur in a haploid cell sometime in the some lower plants and some social insects like a honeybee. It involves major reorganization of the cell component. Okay, so this is the very important phenomena of a mitosis. Mitosis can be divided into two phases. First is a karyokinesis and second is a cytokinesis. So what is the karyokinesis? Karyokinesis is the process of a division of a nucleus, and cytokinesis is a process of a division of a cytoplasm. So first of all, let me discuss about karyokinesis. So if you are talking about karyokinesis, karyokinesis itself can be divided into four stages. So first stage of any karyokinesis will be prophase. Okay, second is the metaphase, third is the anaphase, and fourth and last one is a telophase. In today's lecture, we will discuss about two phases of a karyokinesis of a mitosis. So first stage is a prophase. Okay, so what happened during prophase? It is the longest phase of a mitosis. Okay, it requires more duration of time. It follows the S and G2 phase, means after the S phase and G2 phase only it can be occur. Okay, and in the AS and G2 phase, already you know very well what happened during AS phase. DNA content will be double, and during the G2 phase all the required material is synthesized. So AS and G2 phases, DNA molecules are intertwined. So now let me discuss about the characteristic features and events of a prophase. What happened during the prophase? So first of all, chromosomal material, that is also called chromatin fibers, are untangled. Okay, these are very interwoven structures and it will be untangled, and what happened, they will condensed out. Okay, and to form mitotic chromosomes, and now chromosome will be formed. They are seen to be composed of two chromatids, already we have discussed, attached together at the centromere. You know very well what is the structure of a chromosome. They may have two chromatids and both the chromatids will attach together by using centromere. Centrosome begin to move towards the opposite ends. You know very well what is the centrosomes. Okay, so perpendicular centromere will be attached together. So centrosome begin to move towards the opposite poles. Okay, this is a very important characteristic of any prophase. Each centrosome radiates out microtubules. Okay, and that is called esters. So here ester formation will occur. Okay, two esters together with spindle fibers, means two esters now together with spindle fibers form mitotic apparatus, and by this way mitotic apparatus will be formed. So esters will be formed, that is actually microtubule structure, and two esters together form a spindle fiber. Okay, and that is called mitotic Understand apparatus. More about the prophase by using this diagram. So first of all, what happens? Centrosome shifting to the opposite pole together. So you know very well centromere is made up of two centrosomes, okay, which is perpendicularly attached together. Okay, so both of them will move towards the opposite end. Okay, assembly of a mitotic spindles. Here you can see ester formation occur. Two esters together form mitotic spindles. Okay, and mitotic we can say apparatus will be formed. Here you can see reddish in color. So assembly of a mitotic spindle will be formed here. Then what happen? Chromosome will be condensed. Okay, the chromatid will be condensed together and form the chromosomes with the two sister chromatids. Here you can see along with the centrosomes, centromeres in the center, nuclear membrane breaks and disappear. Another very important phenomena occur here will be a nuclear membrane will be disappear. Okay, or breaks and disappear. And finally, last stage in which last phenomena nucleolus will disappear. So this is the characteristic feature of any prophase. How you can identify? If you observe that two centrosomes will be moved towards opposite end, that is the indication of a prophase. Second indication is what? A chromosome formation. Otherwise what happen? It is an interwoven structure. Okay, you cannot definitely see the or observe the chromosomes in the nucleus, but what happened due to condensation? Okay, what happened? The chromosome formation occur and two sister chromatid will join together by using centrosomes, centromere. Then nuclear membrane breaks. Here you can see nuclear membrane will be break and disappear. This is the third event. And fourth event you will observe is a nucleolus will disappear. These all are the events. By using or observing that you can understand these stage is under the means these cell under the prophase stage. Now let me understand more about the prophase by using this diagram. So here you can see this is the centriole which moves towards the opposite end. Okay, and here esters are forms. Okay, this is in the periphery and the both type of centriole estrels are forms. Okay, that is a mitotic apparatus, and by using that you can see that will be elongated, and that is one kind of a microtubules. As this centriole move towards opposite end, exactly when they reach to the ends, you can see here a nuclear membrane will be disappear, break down and disappear. Here you can see. So now this chromosome will be directly come in contact with the esters. Okay, or spindle fibers which are formed on the centrosomes. And finally, when nuclear membrane disappear, this chromosome will come in contact with this spindle fiber. And here you know very well on the chromosome they are kinetochores. So on the kinetochore, the spindle fiber will be attached. Okay, so by this way all the chromosome will be attached with the spindle fiber. Here you can see. Okay, this is a kinetochore. So kinetochore here play very important role in the cell division process. Now let me summarize whole process. Here you can see central will be shifted to the opposite pole. Then chromosomal condensation occur. Okay, and finally it will be disappear the nuclear membrane. Also a disappearance of nucleolus will be observed during this phase. Okay, and this is means you can observe chromosomes like this. Okay, so you can see two sister chromatids are attached by using a centromere. Okay, so this is the indicative event of any end of any prophase is considered as a late prophase. So during this phase, what you can observe, cells at the end of a prophase do not show Golgi complexes, endoplasmic reticulum, nucleolus, and nuclear envelope. So you cannot observe all these structures at the stage late prophase. As all chromosome attached with the spindle fibers of a centriole or a mitotic apparatus, that all chromosome will arrange like this in the center of a cell, and that is the indication of a initiation of a next stage, and that is a metaphase. Let me discuss about metaphase, that is the second step of a karyokinesis of a mitosis. Okay, so during which what happened? The nuclear envelope completely disintegrate, hence the chromosome spread through the cytoplasm of the cell. Okay, and chromosome condensation is completed. Okay, and as a result of that you can study chromosomes structure very easily, and that's why so many scientists prefer metaphase stage for study purpose of the chromosome. So metaphase stage is very important or very well known for the study of chromosomes, because here in this stage chromosome will be clearly appear because of a disappearance of a nuclear membrane. They can be studied easily under the microscope. They will have two sister chromatids. Here you can see they will have two sister chromatids. Chromosome come to lie at the equator. So here you can see, okay, on the equatorial region that will be arranged. Okay, the plane alignment of the chromosome at the metaphase is called metaphase plate. Okay, so arrangement of the chromosome on the equatorial region is called metaphase plate. The spindle fiber from both the poles are connected to the chromatids by the kinetochore. As I told you already, the spindle fiber which are generated by the centriole, that is actually microtubule, will be connected with the kinetochore present on the chromosome, especially in the centromere of the chromosome. Here you can see whole process of a metaphase. As the spindle fiber generate, all chromosome will be arranged in the metaphase plate, that is equatorial region. Okay, and these centromere will be have a kinetochore, two number of kinetochore on that, and the spindle fiber can only attach on the kinetochore. What is a metaphase? In the metaphase, all the chromosome will be arranged on the equatorial region, that is a metaphase plate. All chromosome arranged on the metaphase plate, next stage will be initiated, and that is so this is your homework for today's lecture. And before we finish this lecture, let me go through one animation video. So look at this video.
Mitosis or karyokinesis, along with cytokinesis, defines the M phase or the mitosis phase of the cell cycle. During mitosis, parent cell divides into two daughter cells, which are not only identical to each other, but also to the parent cell. Mitosis is also known as equational division, as both the parent and the daughter cells have the same number of chromosomes. Mitosis is a continuous process. However, for convenience, it is divided into 4 stages of nuclear division, namely prophase, metaphase, anaphase, and telophase. Prophase is the first phase of mitosis and follows the S and the G2 phases of interphase. During the S phase, the DNA synthesizes or replicates, and in the G2 phase, the cell continues to grow, but the DNA molecules formed are intertwined and not distinct. However, during prophase, chromatin condensation or the untangling of the chromosomal material takes place, and the centriole that had duplicated during the S phase starts to move towards the opposite poles of the cell. Certain characteristic events mark the completion of prophase. Compact mitotic chromosomes are formed due to the condensation of chromosomal material, and each chromosome is made up of 2 chromatids attached together at the centromere. Moreover, the centrioles that had moved to the opposite ends of the cell initiate the formation of the mitotic spindles with the help of the microtubules and the proteinaceous constituents of the cell cytoplasm. At the end of prophase, the nucleolus, endoplasmic reticulum, Golgi complexes, and the nuclear envelope are not visible when viewed under a microscope. Prophase is followed by the metaphase, the onset of which is marked by the disintegration of the nuclear membrane, resulting in the scattering of the chromosomes in the cytoplasm. During this stage, the chromosomes can be clearly seen and studied under the microscope as their condensation is complete. Moreover, each chromosome is made up of 2 sister chromatids bound together by a centromere. Each centromere has a small disc-shaped structure on its surface called the kinetochore. Spindle fibers attach to the kinetochores of the chromosomes, and the chromosomes move towards the spindle equator. The plane of alignment of the chromosomes during this phase is known as the metaphase plate. Metaphase is followed by anaphase.
In next lecture, we will learn next 2 stages, and that is anaphase and telophase. So till that, goodbye.